Welcome, deep divers. Today, we're plunging into a topic that, despite its microscopic size, is absolutely fundamental to life, health, and, well, even some pretty terrifying moments in history.
Hmm.
To sort of set the scene, let's go back to March 20th, 1995. It was morning rush hour, Tokyo subway. A religious cult released sarin, a chemical weapon, into the air.
A terrible event.
Thousands struggled to breathe. And while only 8 people died, hundreds were seriously injured.
Sarin is a small but incredibly deadly molecule. It's a nerve gas, specifically designed to interfere with your nervous system.
How does it work exactly?
Its mechanism is chillingly precise. It basically poisons an enzyme. This enzyme is responsible for breaking down the chemical signals your nerves use to activate muscle cells.
Oh,
So
okay.
without that enzyme working, these signals just build up, leading to paralysis, especially of the muscles you need for breathing.
And that devastating real-world event brings us directly to our mission today. This deep dive is all about the plasma membrane, that incredible boundary surrounding every single cell in your body.
Right.
We're going to uncover how proteins within this very membrane are, like master translators. They convert those vital chemical signals into the electrical signals that power everything from your heart beating to your brain thinking.
Exactly.
Our goal is to extract the most important insights from our source material, giving you a crystal clear understanding of this dynamic cellular boundary without overwhelming you. Let's truly dive in.
What's truly remarkable, I think, is how our understanding of this fundamental structure has evolved over time. It started with some incredibly clever, yet maybe somewhat flawed, early detective work.
Absolutely. Think back to 1925. Two Dutch scientists, E. Gorder and F. Grendel, made a groundbreaking proposal.
Right.
They suggested that cellular membranes might be made of a lipid bilayer, which was, a pretty radical idea at the time.
It really was. And they focused on human red blood cells, which turned out to be perfect for this kind of study.
Why red blood cells specifically?
Well, because they don't have internal organelles or a nucleus. So any fats, any lipids they extracted had to come from the cell's outer skin, the plasma membrane.
Makes sense.
They then spread these lipids out on water, kind of like oil on a puddle, and measured how much surface area they covered.
A simple idea, but powerful. And their conclusion?
Their conclusion was that those red blood cells contained enough lipid, enough fat, to form a layer 2 molecules thick, a double layer, a lipid bilayer.
Wow.
They even figured out that the water-loving parts, the heads, would face outwards toward the water, which is spot on.
But you mentioned flawed.
Ah, yes, here's the fascinating twist. They actually made a couple of experimental mistakes in their measurements. But those errors, almost, serendipitously, canceled each other out.
No way. So they got the right answer for the wrong reasons.
Kinda, yeah. They confirmed the idea of a double layer of lipids, with those water-loving heads out and the water-hating tails tucked inside. It's a great story in science.
So by the 1920s and 30s, this lipid bilayer idea was gaining traction. But then scientists started noticing things that didn't quite fit a simple fat layer model.
Right. Evidence started mounting. For instance, how easily a substance could cross the membrane wasn't only about whether it dissolved in fat. Other factors were involved.
Okay.
And when they measured the surface tension of membranes, it was much lower than what you'd expect from pure lipid structures.
Suggesting something else was there.
Exactly. This was a big clue that something else was present, reducing that tension. Proteins, most likely.
Which leads us to the modern view.
Pretty much. The understanding we have today. Cellular membranes are dynamic structures. You have individual protein molecules actually embedded within, or even standing across, that fluid lipid bilayer.
Fluidity is key then.
Absolutely. It's this beautiful fluidity that makes membranes so adaptable, allowing their various components to move around and interact constantly. It's not a rigid wall.
So we're really talking about these incredible lipid protein assemblies. Lipids for structure, proteins for the specific jobs.
That's a great way to put it. If we zoom out, the lipid bilayer acts primarily as the cell's protective wall and, a barrier, controlling what gets in and out.
Right, the gatekeeper.
But it's the membrane proteins that perform most of the cell's specific tasks. Think about it. A nerve cell, a muscle cell, a skin cell. Each has its own unique set of membrane proteins that enables its specialized functions.
It's amazing customization, and what a diverse cast of characters these membrane components are. Let's talk more about the lipids first.
Okay.
Phospholipids are definitely the core players, right? Forming that essential bilayer structure we talked about.
Yes. They're the most abundant. There are several types, like phosphatidylcholine or phosphatidylserine, but they all share that basic structure, hydrophilic head, hydrophobic tails that makes them spontaneously form the bilayer in water.
And then you mentioned sphingolipids. How are they different?
They're structurally a bit different from the main phospholipids. They often have longer, more saturated fatty chains. This gives them slightly different packing properties within the membrane, maybe making certain areas a bit less fluid.
Interesting. And what about glycolipids? You mentioned they were crucial, but maybe a bit mysterious.
Yeah, their roles are still being fully worked out, but we know they're vital. For instance, your nervous system is particularly rich in certain glycolipids, like galactocerebroside. It's a major component of the myelin sheath that insulates nerve cells.
And the real-world impact is significant, right, if something goes wrong?
Absolutely. A lack of the enzymes needed to build specific glycolipids can lead to devastating neurological diseases. Things like muscular tremors, paralysis, seizures, even blindness. It shows how critical these molecules are.
Wow.
They also play a critical role, unfortunately, in infectious diseases. Many toxins, like those causing cholera and botulism, and even the influenza virus, obviously bind to these glycolipids on your cell surface as a way to get inside.
So they're like docking sites for invaders sometimes.
In a way, yes. It highlights their subtle, yet incredibly powerful influence on cell function and health.
Okay, and we can't forget cholesterol. I read it can make up nearly half the lipids in some animal cell membranes.
That's right, up to 50% in some cases. It's a fascinating molecule. It tucks itself into the bilayer. Its small, water-loving hydroxyl group faces out, but the rest, those flat, rigid rings, are embedded deep within the hydrophobic core.
What does it do there?
Those rigid rings of cholesterol act like tiny stiffeners. They interfere with the wiggling fatty acid tails of the phospholipids nearby.
So it makes the membrane less flexible.
It effectively stiffens the bilayer while maintaining its overall fluidity. It prevents the membrane from becoming too fluid at high temperatures and too rigid or crystalline at low temperatures. It's a buffer, maintaining optimal fluidity.
A truly remarkable balancing act. And it's incredible how such a thin structure, only about 6 nanometers thick, can do so much.
Indeed, it's astonishingly thin. And this thinness, combined with the self-organizing nature of lipids, means that membranes are always continuous and unbroken. Those hydrophobic tails are never exposed to water.
So they form networks.
Exactly. This allows them to form vast, interconnected networks throughout the cell, enclosing compartments and connecting different parts.
And because of that amazing flexibility, membranes are constantly changing shape, aren't they?
Constantly. Think about how cells move, crawling along surfaces, or how a single cell dramatically divides into 2 new cells during division. The membrane is deforming and reshaping every second.
Right.
This deformability is also essential for processes like regulated fusion and budding, like when tiny sacs of material called vesicles fuse with the plasma membrane to release their contents outside the cell, or when 2 cells, like a sperm and egg, merge during fertilization.
And let's connect this basic understanding of phospholipids to something, well, really practical and life-saving. Liposomes.
Yes, liposomes. It turns out, if you just put phospholipids in water in a test tube, they'll spontaneously self-assemble into these tiny, fluid-filled spherical vesicles.
Just on their own.
Just on their own. It's driven by thermodynamics, hiding those hydrophobic tails. These self-assembling structures have been invaluable in research. They let scientists study membrane proteins in a much simpler, controlled environment than a whole cell.
Okay, that's useful for research, but you said life-saving.
Right. Here's the remarkable leap. Scientists realized they could use these liposomes as tiny vehicles to deliver drugs or even DNA molecules inside the body. Imagine a microscopic bubble carrying a specific cargo directly to where it's needed.
Like a targeted delivery system.
Exactly. Early attempts often failed because our immune system is very good at recognizing and destroying foreign particles, including simple liposomes.
So how do they fix that?
They developed stealth liposomes. These have protective outer coating, often a synthetic polymer like polyethylene glycol, that literally hides them from immune cells. A great example is Calyx, a chemotherapy drug used for metastatic breast cancer. It's packaged in a stealth liposome. This allows it to circulate longer and potentially accumulate more in tumor tissues. Some even go a step further.
How so?
By adding targeted antibodies to the liposome surface. These antibodies recognize specific proteins found only on, say, cancer cells. So the liposome delivers its drug cargo much more specifically to the target cells, minimizing side effects.
That's incredible. Biology turned directly into medicine. Speaking of complexity, you mentioned earlier that the two layers, the leaflets of the lipid bilayer, aren't identical.
That's right. They have distinctly different lipid compositions. It's called membrane asymmetry.
How do we know that?
To some clever experiments. For instance, researchers treated intact red blood cells with enzymes that digest lipids but couldn't get inside the cell. By seeing which lipids were digested, they could figure out which ones were predominantly on the outer leaflet.
And what did they find?
They found, for example, that lipids like phosphatidylcholine, PC, are more concentrated in the outer layer, while others, like phosphatidylphenolamine, PE, and especially phosphatidylserine, PS, are found mainly in the inner layer facing the cytoplasm.
And this asymmetry isn't just random, right? It has functions.
Absolutely vital functional significance. Glycolipids, for instance, are exclusively found on the outer leaflet, where they often act as cell surface receptors for signaling molecules or even pathogens, as we discussed.
Okay.
PE in the inner leaflet, with its smaller head group, helps promote membrane curvature, which is important for processes like vesicle budding.
And phosphatidylserine, PS.
PS is really interesting. It's normally kept strictly on the inner leaflet and carries a negative charge, which helps bind positively charged proteins inside the cell. But if PS appears on the outer surface of, say, aging lymphocytes or activated platelets, it acts as a signal.
A signal for what?
For aging cells, it's an eat-me signal, marking them for destruction by macrophages. On platelets, it triggers blood coagulation, so its location is critical. Also, phosphatidylcholine PI lipids, mainly on the inner leaflet, can be phosphorylated and play key roles in signal transduction.
It's a highly organized and functional boundary then, not just a passive bag.
Precisely. Now, moving from the lipids to the protein players. The membrane's functional sidedness is absolutely critical here too. Every membrane protein has a defined orientation relative to the bilayer.
Meaning one side is different from the other.
Very different functionally. This ensures that processes happen in the correct direction. Think of it like a one-way valve or a directional antenna.
Okay. And how do we classify these proteins based on how they sit in the membrane?
We generally group them into three main classes based on their relationship with the lipid bilayer. First, you have the integral membrane proteins.
These are the ones embedded right in.
Yes, they penetrate into the hydrophobic core or even span the entire bilayer. They're tightly associated and are amphipathic.
Amphipathic meaning?
Meaning they have both hydrophilic, water-loving parts that interact with the aqueous environment on either side, and hydrophobic, water-hating parts that sit comfortably within the oily interior of the membrane.
And what do they do?
Oh, a huge range of things. They act as receptors that bind external signals. They form channels or transporters to move specific substances across the membrane. They're involved in energy transfer. The list goes on.
They sound really important.
Immensely important. In fact, integral membrane proteins account for about 25–30% of all the proteins encoded by our genes. And incredibly, they are the targets for roughly 60% of all current prescription drugs.
Oh, that's huge.
It really highlights their central role in physiology and disease. Their structure is fascinating, too. The transmembrane parts are typically alpha helices made of about 20, mostly nonpolar amino acids that fit nicely into the hydrophobic core.
How do scientists even see these? They're so small.
It requires really sophisticated techniques. X-ray crystallography has given us some beautiful structures like the photosynthetic reaction center. And more recently, cryo-electron microscopy, or cryo-EM, has been revolutionary, allowing us to see the structures of large, complex membrane proteins like ion channels in near atomic detail.
Amazing technology. Okay, so that's integral proteins. What's the second class?
The second class are peripheral proteins. These are located entirely outside the lipid bilayer, either on the outer surface or the inner cytoplasmic surface.
How are they attached?
They're associated with the membrane indirectly, usually through non-covalent bonds with integral membrane proteins or with the polar head groups of the lipids. They're more loosely attached than integral proteins.
Okay. And the third type?
The third type are lipid-anchored proteins. These are also located outside the bilayer, but they are covalently linked to a lipid molecule, like a fatty acid or prenyl group, that is embedded within one of the leaflets of the membrane.
So they're tethered, essentially.
It's exactly, tethered by a lipid anchor. Important signaling proteins like CRC and ROS, which are implicated in cell growth control and cancer when they malfunction, are examples of this type.
Got it. Now let's talk about something you hinted might be a bit controversial. Lipid rafts.
Ah, yes, the lipid rafts. This is a concept that, well, it divides cell biologists to some extent.
What's the basic idea?
The idea is that the membrane isn't uniformly fluid, Certain lipids, especially cholesterol and sphingolipids, tend to cluster together, forming small, dynamic microdomains. These domains are thought to be slightly thicker and more ordered, less fluid than the surrounding bilayer.
Like little islands floating in the membrane sea.
Kind of like that, yeah. And the idea is that certain types of proteins, like GPI-anchored proteins or some signaling molecules, preferentially partition into these rafts.
So why the controversy? Sounds plausible.
Part of the challenge is the evidence. Much of the early evidence came from experiments using detergents at cold temperatures to isolate detergent-resistant membranes. Critics argued these might be artifacts of the experimental procedure, not necessarily reflecting what happens in a living cell at normal temperatures.
Okay.
Also, these rafts, if they exist in living cells, are thought to be very small, maybe tens to hundreds of nanometers across, and very transient, perhaps lasting only milliseconds or seconds. This makes them incredibly difficult to observe directly and definitively in real time within a living cell.
So they're hard to pin down.
Very. But the appeal of the lipid raft concept is strong. The membrane can seem like a very random, disorganized sea of molecules. Rafts offer a way to introduce local order.
How so?
They could act as floating platforms that concentrate specific proteins together, perhaps organizing components of a signaling pathway or facilitating interactions needed for certain functions, like membrane budding or signaling cascades.
So a potential organizing principle, but still debated.
Exactly. Whether they exist precisely as originally envisioned or not, it's clear the membrane isn't homogeneous. But regardless of rafts, what is undeniable is the incredible dynamic movement within the membrane. It's a constantly bustling environment.
Let's talk about that movement. Lipids first.
Okay, for lipids, lateral diffusion moving sideways within their own leaflet is incredibly rapid. A single phospholipid molecule can diffuse the length of a bacterial cell, about a micron, in just a second or two.
Wow, that's fast. But what about crossing to the other side?
That's very different. Moving from one leaflet to the other, a flip-flop, is extremely slow for most phospholipids. We're talking hours or even days for spontaneous flip-flop.
Why so slow?
Because the hydrophilic head group of the phospholipid has to somehow pass through the extremely hydrophobic, oily core of the bilayer. That's a huge energy barrier.
Makes sense. But we know the leaflets are asymmetric, so lipids must get sorted somehow.
Yes, cells have specific transporter proteins called flippases, floppases, and scrambloses that actively catalyze the movement of certain lipids between leaflets. Flippases, for example, often move specific lipids like PS and PE from the outer to the inner leaflet, helping establish and maintain that crucial asymmetry we discussed.
Okay, so lipids move laterally fast, but flip-flopping is slow unless helped. What about proteins? How do they move?
Proteins also move laterally, but generally much slower than lipids, and their movement can be much more variable and restricted. Scientists use clever techniques like FRAP, fluorescence recovery, after photobleaching to measure this.
How does FRAP work?
You label the membrane proteins with a fluorescent molecule. Then you use a focused laser beam to irreversibly bleach the fluorescence in a small spot on the cell surface. Then you watch and measure how quickly unbleached fluorescent proteins from the surrounding area diffuse into that bleached spot.
And the speed of recovery tells you how fast they're moving.
Exactly. The faster the fluorescence recovers, the faster the proteins are diffusing.
What did FRA experiments show?
They showed a couple of key things. First, membrane proteins in living cells typically move 10 to 100 times slower than they would if they were in an artificial, pure lipid bilayer. Second, a significant fraction of proteins, sometimes 30 to 70%, depending on the protein and cell type, appeared to be immobile or only capable of diffusing within a very confined area. They don't recover into the bleached spot over the timescale of the experiment.
So something is holding them back.
It seems so. Although FRAP has limitations, it can't easily distinguish between truly immobile proteins and those just diffusing very slowly, or within a small corral. Newer techniques like single particle tracking or using optical tweezers have given us more insight.
Optical tweezers, that sounds like science fiction.
It's pretty amazing. You can attach a tiny antibody-coated bead to a specific membrane protein. Then you use a highly focused laser beam, the optical tweezers, to physically grab the bead and try to drag the protein through the membrane.
And what does that reveal?
By measuring the force needed to move the protein, or observing where it gets stuck, you can map out barriers and corrals within the membrane. These studies confirm that protein movement is often restricted.
So what restricts their movement? What are these barriers?
Several things seem to be involved. The cytoskeleton, the cell's internal protein scaffold lying just beneath the plasma membrane, plays a big role. Filaments of the cytoskeleton can attach to integral membrane proteins, anchoring them or creating fences that confine other proteins to specific domains.
Like pens keeping sheep in.
Exactly like that. Also, extracellular matrix components outside the cell can interact with membrane proteins and hinder their movement. And finally, interactions with other integral membrane proteins can form large complexes or clusters that diffuse very slowly or act as barriers themselves.
It's a crowded and organized environment then, even for lipids.
Interestingly, yes, even phospholipid movement isn't completely random. High-resolution tracking shows that even lipids can be transiently confined within small compartments for fractions of a second before hopping over a fence into an adjacent compartment. It's thought these fences might be rows of integral proteins anchored to the underlying cytoskeleton.
All this leads to the idea of membrane domains and cell polarity, right? The membrane isn't uniform.
Absolutely not. Membranes exhibit distinct variations in protein composition and mobility across different regions of the same cell. This is crucial for cell function, especially in polarized cells.
Like what kind of cells?
A classic example is epithelial cells, like those lining your intestine or kidney tubules. They are highly polarized. They have an apical surface facing the lumen, the inside of the intestine or tubule, lateral surfaces contacting neighboring cells, and a basal surface facing the underlying tissue.
And each surface is different.
Very different. Each domain has a unique set of membrane proteins, transporters, receptors, adhesion molecules tailored to its specific function, like absorbing nutrients from the gut lumen or pumping ions into the bloodstream. This compartmentalization is essential.
Any other examples?
Sure, think about a highly differentiated cell like a sperm cell. It's covered by a single continuous plasma membrane, but if you map out the location of specific proteins, you find a striking mosaic. Different proteins are precisely localized to the head, mid-piece, or tail regions, reflecting their specialized roles in fertilization.
So the membrane is a dynamic mosaic locally specialized for function.
That's a perfect summary. This compartmentalization allows specific proteins, like receptors for neurotransmitters or hormones, to be concentrated precisely where they're needed.
Okay, so we've explored the structure, the components, the dynamics, but now let's get to one of the most critical functions. How do things actually get across this complex barrier? Let's talk membrane transport.
Right. And to understand this, scientists often turn back to our old friend, the human red blood cell plasma membrane. It really is the quintessential model system for membrane transport studies.
Why is it so good for this?
Well, as we said, they're cheap. You can get huge numbers of them easily. They're already single cells, so no complex tissue dissociation is needed. And critically, they're simple internally. They lack internal membrane-bound organelles, like mitochondria or nucleus.
So the only membrane you're dealing with is the outer plasma membrane.
Exactly. And you can easily isolate large quantities of pure plasma membranes. You place the cells in a hypotonic solution water with very low solute concentration. Water rushes in via osmosis. The cells swell up and burst, releasing their hemoglobin and other cytoplasmic contents. What's left behind are empty membrane sacs called ghosts.
Pure plasma membrane ghosts, ready for study.
Perfect for studying transport proteins without interference from other cellular components.
Okay, so using systems like this, what have we learned? How do substances move across?
There are basically 4 fundamental mechanisms by which substances cross cellular membranes.
4 ways. Let's break them down.
Okay. First, the simplest. Simple diffusion through the lipid bilayer itself.
Just passing right through the fat.
Essentially, yes. Movement is always passive, meaning it doesn't require the cell to expend energy, and it always goes down the concentration gradient from high concentration to low concentration.
What determines how easily something diffuses this way?
2 main factors: lipid solubility and size. The more lipid-soluble hydrophobic a substance is, the more easily it can dissolve in and pass through the membrane's core. And, all else being equal, smaller molecules penetrate faster than larger ones.
Can you give some examples?
Sure. Very small, uncharged molecules like oxygen, O2, carbon dioxide, CO2, nitric oxide, NO, and even water, H2O, although polar is small enough to penetrate rapidly via simple diffusion. But larger polar molecules like sugars, amino acids, and especially charged ions penetrate the lipid bilayer very poorly, if at all. The bilayer is an excellent barrier to these substances.
Keeping the important stuff inside. Okay, what's the second way?
The second way is simple diffusion through an aqueous channel. These are protein-lined pores through the membrane.
Like a tunnel.
Exactly. These channels are typically selected for certain ions or small molecules, like water. aquaporins. Again, movement is passive down the concentration gradient, but it bypasses the lipid bilayer entirely.
Got it. Method 3.
Method 3 is facilitated diffusion. This also involves a membrane protein, but instead of a simple channel, it's a transporter or carrier protein.
How does that work?
The solute molecule, like glucose or an amino acid, binds to a specific site on the transporter protein. This binding triggers a conformational change, a shape change in the protein, which exposes the solute to the other side of the membrane where it's released.
Is energy needed?
No, it's still passive.
Yeah.
Net movement only occurs down the concentration gradient. The protein just facilitates or helps the movement across the barrier for molecules that can't easily diffuse on their own. Think of it like a revolving door versus an open tunnel.
Can you give an example?
The classic example is the family of glucose transporters, GLUT proteins. These proteins move glucose from your bloodstream, where its concentration is usually higher after a meal, into your cells, where it's lower because it's being used for energy. Insulin actually promotes this process in many tissues by causing more GLUT transporters to be inserted into the plasma membrane.
Okay, passive diffusion, channels, facilitated diffusion. What's the fourth way?
The fourth way is active transport. This is the only one of the four that can move substances against their concentration gradient from low concentration to high concentration.
Like pushing something uphill?
Precisely. And just like pushing something uphill requires effort, active transport always requires the cell to expend metabolic energy, usually derived from the hydrolysis of ATP. These systems involve protein pumps.
So 4 ways: simple diffusion, bilayer or channel, facilitated diffusion, and active transport.
That's the basic toolkit.
Yeah.
Now let's quickly touch on the movement of water specifically, which is governed by osmosis.
Right, you mentioned osmosis earlier with the red blood cell ghosts.
Exactly. Membranes are considered semi-permeable because water molecules generally move across them much more rapidly than most dissolved solutes, like ions or sugars. Osmosis is defined as the net movement of water across a semi-permeable membrane from a region of lower solute concentration to a region of higher solute concentration.
Water moves to dilute the side with more stuff dissolved in it.
That's the gist of it, yes. It's driven by the difference in water concentration, or more formally, water potential.
And this has real effects on cells.
Huge effects. If you place a cell in a hypotonic solution, lower solute concentration than inside the cell, water will rush in, causing the cell to swell and potentially burst. Like the red blood cells we discussed, it's called hemolysis.
Okay.
If you place it in a hypertonic solution, higher solute concentration, water will flow out, causing the cell to shrink and shrivel.
And isotonic.
In an isotonic solution, the solute concentration is the same inside and out, so there's no net movement of water, and the cell maintains a stable volume. This is crucial for maintaining cell integrity in our bodies. Osmosis is also vital for things like fluid reabsorption in your digestive tract and kidneys. In plants, it generates turgor pressure, which keeps them rigid. Wilting is basically water loss due to osmosis.
Makes sense. Now let's get back to those ions. You said the bilayer is highly impermeable to them, but their movement is critical. How does that happen?
That's where ion channels come into play. These are the aqueous channels we mentioned as the second transport mechanism, but they deserve special attention because they are so critical for nerve and muscle function, among other things.
So these are specific protein tunnels for ions.
Exactly. Ion channels are integral membrane proteins that form a hydrophilic pore through the membrane. They are typically highly selective, meaning a potassium channel mainly lets potassium ions through, a sodium channel mainly lets sodium through, and so on. And movement through them is always passive diffusion down the electrochemical gradient.
But they aren't always open, are they?
Correct. Most ion channels are gated, meaning they can exist in either an open or a closed conformation. The transition between these states is regulated.
What regulates?
There are 3 main types of gating mechanisms. Voltage-gated channels open or close in response to changes in the electrical potential across the membrane. Ligand-gated channels open or close when a specific molecule, a ligand like a neurotransmitter, binds to the channel protein. And mechanogated channels respond to physical forces like stretching or pressure on the membrane important for touch and hearing.
Voltage-gated channels sound particularly important for nerves.
They are absolutely essential. And here's a really fascinating story about the voltage-gated potassium ion channels. For a long time, we knew they existed and were crucial, but understanding how they worked at a molecular level was a huge challenge.
What was the breakthrough?
In 1998, Roderick McKinnon and his colleagues published the first atomic resolution structure of a bacterial potassium channel, called KCSA, determined by X-ray crystallography. It was a landmark achievement.
What did this structure reveal?
It showed the incredible elegance of its design. The channel has a narrow region called the selectivity filter. It's lined by precisely arranged carbonyl oxygen atoms from the protein backbone. These oxygens perfectly mimic the way water molecules normally surround a potassium ion, K+, allowing the K+ ion to shed its water shell and pass through the filter.
How does it exclude sodium ions, Na+? They're smaller, aren't they?
Sodium ions are slightly smaller, yes, but they hold on to their surrounding water molecules more tightly. The selectivity filter is just the right size to coordinate a dehydrated K+ ion, but it's too large to effectively coordinate the smaller Na+ ion in a way that compensates for the energy needed to remove its water shell. So Na+ ions typically can't pass through. It's an exquisite molecular sieve.
That's amazing. And they move fast?
Incredibly fast. The structure suggested that K+ ions move through the filter almost like billiard balls, multiple ions occupying sites within the filter simultaneously, pushing each other through with very little energy barrier. This allows for rates approaching the theoretical limit of diffusion. Eukaryotic voltage-gated potassium channels, like the famous Shaker channel studied in fruit flies, operate on similar principles, with additional domains that sense voltage changes and open or close the pore.
Incredible design. Okay, let's shift to the energy-consuming side. Active transport pumps. The big one is the sodium-potassium pump, right?
Definitely. The NAT plus K plus AT pace, as it's formally known, is a cornerstone of animal cell physiology. It's a primary active transport system, meaning it directly uses the energy from ATP hydrolysis to move ions.
What is move?
For every molecule of ATP it breaks down, it pumps 3 sodium ions, NAT plus, OT of the cell, and 2 potassium ions, K plus, into the cell. Both of these movements are against their respective concentration gradients.
3 sodium out, 2 potassium in. That doesn't seem electrically balanced.
It's not. Because it moves 3 positive charges out for every 2 positive charges in, the pump is electrogenic. It contributes directly to the electrical potential difference, voltage, across the membrane, making the inside slightly more negative.
What kind of pump is it?
It belongs to a large family called P-type ion pumps, characterized by being phosphorylated, having a phosphate group temporarily attached as part of their reaction cycle.
And why is this pump so important? What does it do for the cell?
It has several crucial roles. First, it helps maintain cell volume by controlling solute concentrations. Second, and perhaps most famously, it generates and maintains the steep concentration gradients for sodium, low inside, high outside, and potassium, high inside, low outside. These gradients are absolutely essential for things like nerve impulse transmission and muscle contraction.
So it sets the stage for electrical signaling.
Exactly. It's constantly working in the background, consuming a significant portion of a cell's energy. Estimates range from 1/3 of the total energy expenditure in many cells up to perhaps 2/3 in active nerve cells.
Wow, that's a lot of energy. Any medical relevance?
Oh yes. The cardiac glycoside drug digitalis, derived from the foxglove plant, has been used for centuries to treat heart conditions. It works by specifically inhibiting the Na plus K plus ATPase in heart muscle cells. This leads to a cascade of effects that ultimately increases calcium levels inside the cells and strengthens heart contractions.
Fascinating connection. Are there other important P-type pumps?
Another well-known one is the H plus K plus ATPase, the proton pump responsible for secreting concentrated acid, HCl, into your stomach. It's found in the parietal cells of the stomach lining.
The stomach acid pump, how is that regulated?
It's cleverly regulated. In resting cells, most of these pumps are stored within intracellular vesicles. But when food enters the stomach, signals, like the hormone histamine binding to its receptor, trigger these vesicles to fuse with the plasma membrane, inserting the pumps into the apical surface, where they can start pumping protons, H+, out into the stomach lumen in exchange for potassium ions, K+.
And this relates to heartburn medications.
Directly. Drugs like Prilosec, omeprazole, and Nexium are proton pump inhibitors, PPIs. They directly block the H plus K plus ATPase enzyme. Other drugs like Zantac, ranitidine, or Pepcid, famotidine, work differently. They are H2 receptor antagonists, blocking the histamine signal that tells the cell to insert the pumps in the first place.
Okay, that covers primary active transport using ATP directly. What was that other type you mentioned?
Secondary active transport, also often called co-transport. This is a really clever mechanism where the cell takes advantage of an existing ion gradient, usually a sodium gradient, established by the Na plus K plus ATPase to drive the transport of another solute against its gradient.
So it uses one gradient's energy to build another gradient?
Exactly. It couples the downhill movement of one solute, like Na, plus flowing into the cell down its steep gradient. to the uphill movement of another solute, like glucose or an amino acid moving into the cell against its gradient.
Can you give the prime example again?
The best studied example is the Na plus glucose cotransporter, found in the epithelial cells lining your intestine and kidney tubules. This protein binds to both sodium ions and glucose on the outside of the cell, where Na plus is high. Because Na plus really wants to flow into the cell down its gradient, the transporter uses the energy released by letting two Na plus ions flow in to also drag one glucose molecule in with them, even if the glucose concentration inside the cell is already much higher than outside.
Hitching a ride on the sodium gradient.
Precisely. It's incredibly efficient. This cotransporter can actually move glucose into the cell against a concentration gradient of more than 20,000-fold.
That's remarkable power. Are there different kinds of cotransport?
Yes. When the two transported species move in the same direction across the membrane, like Na plus and glucose both moving in, it's called symport. When the two species move in opposite directions, for example, one moves in while the other moves out, it's called antiport, or exchange. A common example of antiport is the Na plus H plus exchanger, which uses the inward Na plus gradient to pump H plus ions out of the cell, helping to regulate intracellular pH.
Okay, that clarifies the main transport mechanisms. Now, shifting gears a bit, let's look at the direct impact when these membrane components, especially channels, malfunction in human health and disease.
This is a really important area. It turns out that mutations in the genes encoding ion channel proteins are responsible for a surprisingly large number of inherited human disorders, often called channelopathies.
What kind of disorders?
The list is long and diverse. It includes certain types of migraine headaches, episodic ataxia, problems with coordination, various forms of inherited paralysis, some types of deafness, certain forms of hypertension, high blood pressure, myasthenia gravis, muscle weakness, kidney stones, and significantly a number of cardiac arrhythmias, irregular heartbeats, that can lead to sudden death.
So defects in these tiny channels can have really severe consequences.
Absolutely. And perhaps the most intensively studied and common inherited ion channel disorder is cystic fibrosis, or CF.
Right, CF. What's the underlying defect there?
CF results from a defect in a specific ion channel found in epithelial cells, the cells that line the airways, sweat glands, pancreas, and other organs.
Which channel is it?
The gene responsible for CF was identified back in 1989. It encodes a protein called the cystic fibrosis transmembrane conductance regulator, or CFTR.
CFTR, what does it do?
Primarily, CFTR functions as a chloride ion Cl channel. Its opening and closing is regulated by cyclic AMP, a common intracellular signaling molecule. However, research has shown it's more complex than just a simple chloride channel.
Oh, how so?
Well, CFTR also appears to conduct bicarbonate ions, HCO3, which is important for controlling the pH of secretions. It also seems to suppress the activity of another channel, the epithelial sodium channel, ENaC, and stimulate other chloride bicarbonate exchangers. So it's kind of a master regulator of ion transport in these epithelia. And how does a defect in CFTR lead to the symptoms of CF?
The main problem, particularly in the lungs, is that the abnormal ion movement across the airway epithelial cells leads to a decrease in the volume of the thin layer of liquid, the airway surface liquid, that normally bathes these cells.
Making the mucus thicker.
Exactly. The mucus layer becomes dehydrated, thick, and sticky. This impairs the function of the cilia, the tiny hair-like projections that normally beat to clear mucus and trapped bacteria out of the airways. The result is mucus obstruction and chronic bacterial infections, leading to progressive lung damage, which is the main cause of mortality in CF.
Such a cascade from one faulty protein. Are there treatments that target this basic defect?
There are treatments aimed at symptoms, like inhaling hypertonic saline solution to try and draw water back into the airway liquid. In terms of targeting the underlying defect, drug development has been intense but challenging. Early attempts to develop drugs that inhibit the ENaC channel, thinking that might compensate for CFTR loss, largely failed due to side effects.
Yeah, so the focus shifted back to CFTR itself.
Right. The most common mutation causing CF, found in about 70% of patients, is called AF508, delta F508. This mutation involves the deletion of a single amino acid, phenylalanine, at position 508. The main problem with the F508 protein is not that it can't function as a channel, but that it misfolds during its synthesis and gets stuck inside the cell, failing to reach the plasma membrane where it's needed.
So it's a trafficking defect.
Primarily, yes, But here's a fascinating discovery. Researchers found that if you grow cells carrying the F508 mutation at a lower temperature, around 30 degrees C instead of 37 degrees C, a significant fraction of the mutant CFTR protein does manage to fold correctly enough to reach the cell surface, and once there, it functions reasonably well as a chloride channel.
Wow. That suggests the potential for correction.
Exactly.
Oh.
That finding spurred a massive effort to screen for small molecule drugs that could either help the F508 protein fold better, called correctors, or help the channels that do reach the surface stay open longer, called potentiators.
And has that led to successful drugs?
It has, though it's been a step-by-step process. An early corrector candidate, VX809, lumacaftor, showed only modest benefit when used alone. A potentiator drug, calideco, avacaftor, was approved first, but it only works for patients with specific, less common mutations, like G551D, where the channel gets to the membrane but doesn't open properly. Calideco helps prop those channels open.
But what about the common A508 mutation?
Here's the exciting part. Clinical trials showed that combining calideco, the potentiator, with VX809, the corrector, provides significant clinical benefit for patients who have 2 copies of the A508 mutation. The idea is that the corrector helps a bit more mutant protein get to the surface, and the potentiator helps those channels that do get there function better. More recently, even more effective combinations involving newer generation correctors have been approved, really transforming treatment for many CF patients.
That's fantastic progress. Before we leave CF, is there any thought on why this mutation is so common, especially in people of European descent?
Yes, there's a hypothesis related to heterozygote advantage. The idea is that carrying 1 copy of the CFTR mutation, being a heterozygote or carrier, might have provided some protection against diseases like cholera or typhoid fever, which cause severe diarrhea due to excessive chloride and fluid secretion triggered by bacterial toxins. Having slightly reduced chloride secretion might have been beneficial against these historically deadly infections.
Interesting evolutionary trade-off. What about gene therapy for CF? Seems like a prime target.
It has been a major goal for decades, especially since the airway cells are relatively accessible via inhalation. Various approaches have been tried.
Like using viruses.
Yes, using modified harmless viruses like adenovirus to deliver a correct copy of the CFTR gene. However, these approaches have faced challenges, including triggering immune responses against the virus and the fact that the correction tends to be transient, as epithelial cells turn over.
What about non-viral methods?
Other approaches have used positively charged liposomes to package the CFTR gene as DNA or mRNA and deliver it to the airway cells. This tends to be safer, avoiding the immune issues of viruses, but historically it's been less efficient at getting the gene into enough cells to make a clinical difference. So far, unfortunately, gene therapy trials haven't yielded significant, lasting improvements for CF patients, but research is definitely ongoing.
A complex challenge. Okay, let's transition now to perhaps the most dramatic display of membrane function, the nerve signal. How does the plasma membrane generate and propagate electrical signals?
It all starts with establishing the resting membrane potential. This is the voltage difference across the plasma membrane of a neuron or other excitable cell when it's not actively signaling.
And how is this potential set up?
2 key players we've already met are involved. First, the Na plus K plus At pace tirelessly pumps Na plus out and K plus in, creating those steep concentration gradients, low Na plus inside, high K plus inside.
Okay, step 1. Establish the gradients.
Step 2 involves ion channels. In a resting nerve cell, the plasma membrane is much more permeable to potassium ions, K+, than to sodium ions, Na+. This is because there are specific types of potassium channels, often called K+ leak channels, like the K2P family, that are mostly open even in the resting state.
So potassium can leak out.
Exactly. Because the K+ concentration is high inside and low outside, K+ ions tend to flow out of the cell through these open leak channels down their concentration gradient.
But potassium ions are positively charged. What happens when they leave?
As positive charges, K+, leave the cell, they leave behind an excess of negative charges inside the cell, mostly trapped anions like proteins and phosphate. This separation of charge creates an electrical potential difference or voltage across the membrane, with the inside becoming negative relative to the outside.
So the chemical gradient pushing K+ out is balanced by an electrical gradient pulling K+ back in.
Precisely. The outward flow of K+ continues until the electrical potential difference becomes negative enough to exactly counteract the tendency for K+ to leave due to the concentration gradient. This point of equilibrium typically results in a resting potential of around negative 70 millivolts MV in a typical neuron. It's like a charged battery storing potential energy.
A battery ready to fire. So how does
Yeah.
it fire? How is the action potential, the nerve impulse itself, generated?
An action potential is triggered when the neuron receives a stimulus perhaps from another neuron at a synapse that causes a small, localized depolarization of the membrane. Depolarization just means the membrane potential becomes less negative. Usually moves from negative 70 milliV towards negative 60 milliV.
What happens if the depolarization is strong enough?
If the depolarization reaches a critical threshold level, typically around negative 50 milliV to negative 55 milliV, it triggers a dramatic stereotyped event involving voltage-gated ion channels.
The voltage-gated channels kick in. Which one's first?
First, voltage-gated sodium channels open very rapidly.
And sodium is high outside, low inside, and the inside is negative.
Exactly. So when these channels open, Na plus ions rush into the cell, driven by both their steep concentration gradient and the negative electrical potential inside.
Flooding the cell with positive charge.
Right. This massive influx of positive charge causes a rapid and dramatic reversal of the membrane potential. It shoots up from the threshold level past zero and typically reaches a peak of about plus 30 to plus 50 millivolts. This rapid depolarization phase is the rising phase of the action potential.
And it happens automatically once threshold is reached.
Yes, it's an all-or-none phenomenon. Subthreshold depolarizations fade away, but once the threshold is crossed, the positive feedback loop of Na plus influx, causing more Na plus channels to open, ensures that a full-blown action potential is generated, regardless of the strength of the initial stimulus beyond the threshold. It's like flushing a toilet, push the handle gently, nothing happens. Push it past the threshold, and the whole tank empties.
Where does the energy for this massive ion movement come from?
That's the beauty of it. The immediate energy for the ion flows during the action potential comes entirely from the potential energy stored in the ion gradients that were established beforehand by Na plus K plus 8T pace. The pump does the slow, steady work of charging the battery. The action potential is the rapid discharge.
A very efficient system. Any medical connections here?
Absolutely. Local anesthetics like Procaine, Novocaine, or Lidocaine work primarily by blocking these voltage-gated sodium channels. By preventing the channels from opening, they block the generation and propagation of action potentials along nerve fibers, particularly those carrying pain signals to the brain. No action potentials, no pain sensation.
Clever. So the action potential fires at one spot. How does it travel down the nerve axon as an impulse?
The action potential doesn't actually travel in the sense of one thing moving. Instead, it's regenerated anew at each adjacent point along the axon membrane.
How does that work?
The large influx of Na plus ions during the action potential at one point creates a local current that spreads passively along the axon for a short distance. This local current depolarizes the adjacent patch of membrane to its threshold.
Triggering a new action potential there.
Exactly. That new action potential then depolarizes the next patch, and so on, creating a wave of depolarization, the nerve impulse that sweeps down the axon. There's also a brief refractory period after an action potential fires, during which the Na plus channels are inactivated and K plus channels are open, preventing the impulse from traveling backwards.
What determines how fast this propagation happens?
Two main factors influence conduction velocity. One is the diameter of the axon. Larger diameter axons offer less internal resistance to current flow, so the impulse propagates faster. Think of it like water flowing faster through a wider pipe.
Okay, diameter matters. What's the other factor?
The other and even more significant factor in vertebrates is myelination.
The fatty sheath around the axon.
Right. Many vertebrate axons are wrapped in a thick, insulating layer called the myelin sheath, produced by specialized glial cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. This sheath is interrupted at regular intervals called the nodes of Ranvier, where the voltage-gated channels are concentrated.
How does myelin help?
Myelin acts as an excellent electrical insulator, preventing ion leakage across the membrane in the myelinated segments. This forces the action potential to essentially jump from one node of Ranvier to the next, a process called saltatory conduction. This jumping is much, much faster than continuous propagation along an unmyelinated axon of the same diameter.
So myelin dramatically speeds things up.
Dramatically. And the tragic real-world consequence of damage to myelin is seen in diseases like multiple sclerosis, MS. MS is an autoimmune disease where the body's own immune system attacks and destroys the myelin sheath in the brain and spinal cord. This disrupts or blocks nerve impulse transmission, leading to a wide range of neurological symptoms like muscle weakness, difficulty walking, vision problems, and fatigue.
A devastating illustration of myelin's importance. Okay, so the impulse reaches the end of the axon. How does it communicate with the next cell, another neuron, or a muscle or gland cell?
This communication happens at specialized junctions called synapses.
Right, and usually the cells don't actually touch, do they?
Correct. In most chemical synapses, the axon terminal of the transmitting neuron, the presynaptic neuron, comes very close to, but doesn't physically touch, the target cell, postsynaptic cell. There's a tiny gap between them, typically 20-50 nanometers wide, called the synaptic cleft.
So the electrical signal can't just jump across. How is the message relayed?
It's relayed chemically using signaling molecules called neurotransmitters. These are stored in small, membrane-bound sacs called synaptic vesicles within the presynaptic terminal.
Walk me through the process.
Okay. 1. When an action potential arrives at the presynaptic terminal, the depolarization opens voltage-gated calcium channels. 2. Calcium ions, Ca2+, rush into the terminal. 3. This influx of calcium triggers the synaptic vesicles to fuse with the presynaptic membrane, releasing their neurotransmitter contents into the synaptic cleft, a process called exocytosis. 4. The neurotransmitter molecules diffuse rapidly across the neurocleft. 5. They bind to specific receptor proteins located on the postsynaptic membrane of the target cell.
And what happens when the neurotransmitter binds to the receptor?
That binding triggers a response in the postsynaptic cell. Depending on the specific neurotransmitter and the type of receptor it binds to, the effect can be either excitatory or inhibitory.
Excitatory meaning?
Excitatory means the binding opens channels, often ligand-gated sodium or cation channels, that allow positive ions to flow into the postsynaptic cell. This causes a small local depolarization called an excitatory postsynaptic potential, EPSP. If enough EPSPs sum up to reach the threshold, the postsynaptic neuron will fire its own action potential.
And inhibitory?
Inhibitory means the binding typically opens channels, often ligand-gated chloride channels or potassium channels, that either let negative ions, Cl, flow in, or positive ions, K+, flow out. This causes a small hyperpolarization, making the inside more negative, or stabilizes the resting potential, making it less likely that the postsynaptic cell will reach threshold and fire an action potential. This is called an inhibitory postsynaptic potential, IPSP.
So each neuron is constantly integrating these incoming signals.
Absolutely. A typical neuron in your brain receives input from thousands of other neurons, some excitatory, some inhibitory. It's constantly summing up these potentials over time and space. Only if the net effect reaches the threshold at the axon hillock where the axon originates will the neuron fire its own action potential. It's a complex decision-making process at the cellular level.
Can you give examples of neurotransmitters and their effects?
Sure. Acetylcholine, ACH, is a classic example. It's excitatory at the neuromuscular junction, causing skeletal muscles to contract, but it's inhibitory at the heart, slowing heart rate. This shows that the receptor determines the effect. In the brain, glutamate is the major excitatory neurotransmitter, while GABA, gamma-aminobutyric acid, is the primary inhibitory neurotransmitter. Many sedatives and anti-anxiety drugs like Valium, diazepam work by enhancing the inhibitory effects of GABA.
It seems crucial that the neurotransmitter signal doesn't last too long. How is it terminated?
Yes, rapid removal or inactivation of the neurotransmitter from the synaptic cleft is vital for allowing precise timing of signals and preventing constant stimulation or inhibition. There are 2 main ways this happens.
What are they?
One way is enzymatic degradation within the synaptic cleft. Acetylcholine, for example, is rapidly broken down by the enzyme acetylcholinesterase. The second major way is reuptake. Specific transporter proteins in the presynaptic terminal, or sometimes in nearby glial cells, actively pump the neurotransmitter back out of the cleft, recycling it for later use. This is common for neurotransmitters like serotonin, dopamine, norepinephrine, and glutamate.
And interfering with these removal processes is how many drugs work, right?
Exactly. This is where it really comes together with pharmacology and medicine. Modulating neurotransmitter levels or actions at the synapse has profound physiological and behavioral effects.
Let's revisit sarin nerve gas from the introduction.
Right. Sarin is a potent inhibitor of acetylcholinesterase. By blocking the enzyme, it causes acetylcholine to build up to abnormally high levels in synapses, particularly at the neuromuscular junction. This leads to continuous muscle stimulation, resulting in convulsions, paralysis, including respiratory muscles, and death. On a related note, milder, reversible inhibitors of acetylcholinesterase are actually used as treatments for Alzheimer's disease, aiming to boost cholinergic signaling, which is deficient in that condition.
What about drugs affecting mood or behavior?
Many work by targeting reuptake transporters. For example, the most common class of antidepressants, the SSRIs, selective serotonin reuptake inhibitors, like Prozac, fluoxetine, and Zoloft, sertraline, work by blocking the reuptake of serotonin, thereby increasing its concentration in the synaptic cleft and enhancing its effects.
And illicit drugs.
Cocaine famously interferes with the reuptake of dopamine, particularly in the brain's reward or pleasure centers, part of the limbic system. This leads to elevated dopamine levels and the associated euphoria. Amphetamines work similarly, both stimulating dopamine release and interfering with its reuptake. Conversely, many antipsychotic drugs used to treat schizophrenia work by blocking certain types of dopamine receptors.
What about marijuana?
The active compound in marijuana, THC, binds to specific receptors called cannabinoid receptors, CB1, which are primarily located on presynaptic terminals in the brain. Binding of THC, or the body's own natural endocannabinoids, to these receptors generally reduces the amount of neurotransmitter release from that terminal upon arrival of an action potential, so it acts as a modulator of synaptic transmission.
Fascinating how these chemicals hijack the synapse.
Huh.
This leads us to the final key concept, synapse and plasticity. Synapses aren't fixed, are they?
Not at all. One of the most exciting areas of neuroscience is understanding how synapses change their strength and efficacy over time based on patterns of activity. This is called synaptic plasticity, and it's believed to be the fundamental cellular mechanism underlying learning and memory.
Where is this studied most intensely?
A major focus has been the hippocampus, a brain region known to be crucial for forming new memories. Researchers discovered decades ago that brief, high-frequency stimulation of certain pathways in the hippocampus can cause a long-lasting enhancement in the strength of the stimulated synapses. This phenomenon is called long-term potentiation, or LTP.
How does LTP work at the molecular level?
A key player in many forms of LTP is the NMDA receptor, a type of glutamate receptor. Under normal conditions, this receptor channel is blocked by a magnesium ion, Mg2+. However, when the postsynaptic membrane is strongly depolarized by intense stimulation, the Mg2+ block is relieved. If glutamate binds to the NMDA receptor while the membrane is depolarized, the channel opens and allows calcium ions, Ca2+, to flow into the postsynaptic cell.
And calcium acts as a signal.
Yes, this influx of Ca2+ acts as a critical second messenger, triggering a cascade of biochemical changes within the postsynaptic neuron. These changes can include inserting more glutamate receptors, like AMPA receptors, into the synaptic membrane, increasing the sensitivity of existing receptors, and even structural changes that strengthen the synaptic connection. The net result is that the synapse becomes more sensitive to future glutamate release that has been potentiated.
So LTP strengthens connections that are actively used together.
Essentially, yes. It allows synapses to become more effective at transmitting signals after periods of high activity. This increased efficiency is thought to be a major way the brain encodes new information and stores memories. Consistent with this, studies show that blocking LTP pharmacologically or genetically often impairs learning and memory formation in animals.
And conversely, problems with synaptic function are linked to disease.
Very much so. It's increasingly recognized that dysfunctions in synaptic transmission or plasticity likely underlie many neurological and psychiatric disorders, including things like myasthenia gravis, affecting the neuromuscular synapse, Parkinson's disease, involving dopamine synapses, epilepsy, imbalances in excitation inhibition, schizophrenia, depression, and anxiety disorders. Understanding the membrane's role at the synapse is key to understanding both normal brain function and disease.
What an incredible journey through the plasma membrane. We started with its basic lipid protein structure, the clever experiments revealing its blueprint.
Explored the diverse cast of lipids and proteins, their dynamic movements, the controversial lipid rafts.
Dove into the mechanisms of transport, from simple diffusion to the sophisticated pumps and channels.
Saw how defects cause diseases like cystic fibrosis, and finally unpacked the membrane's starring role in generating nerve impulses and mediating communication at the synapse.
This deep dive has truly revealed the plasma membrane, not just as a static boundary, but as an incredibly dynamic, intelligent, and responsive hub, constantly interacting, communicating, and adapting. It's absolutely fundamental to life itself.
So as we finish, here's something to ponder. Given the complexity and dynamism we've uncovered, what other secrets do these cellular boundaries hold? And how might unlocking those secrets further transform medicine and indeed our very understanding of life? Thank you, as always, for joining us on this deep dive. We really appreciate you being a part of our deep dive family.